Okay, before we get started, let's do a little review here. In which way is a genomic island different from the rest of an organism's chromosomal DNA? Is it A, genomic islands are separate DNA molecules? B, chromosomal DNA contains more proteins than a genomic island? C, chromosomal DNA is much smaller than the DNA of a genomic island? Or D, genomic islands have a different percentage of GC base pairs than chromosomal DNA? Go ahead and pause the video, think about this one. All right, the answer would be D, right? They have a different GC percentage. These other ones do not make sense. Genomic islands, different GC because they were probably acquired through horizontal gene transfer. In Chapter 18.2, we're going to talk about the first steps in the process, pathogen entry and then pathogen colonization. We're going to talk about why pathogens need to attach to the host cells, did a little overview of that, but we'll talk about ways that they do this. And then we'll particularly talk about pili. We have two types, type 1 and type 4. And then we'll talk about something really critical, biofilms, which are super important to pathogenicity. Okay, so like I said, your human body has lots of mechanisms that try to get rid of pathogens, get rid of microbes. The mucociliary escalator in the lungs, right? Those cilia beating to bring all that phlegm and junk up out of your lungs. Peristaltic motion in your intestines, constantly pushing things through. Urine flow out of the urethra and the bladder, trying to dislodge any pathogens that may come in there. These all push microbes out. And if pathogens didn't have ways to attach to the cell surface, they would quickly be rid from the body. We call any factor that promotes attachment an adhesin. This is a very general term. There's lots of different types of adhesins. We have some like pili or fimbriae, if you hear that term. We will see pili as one type. Then there's a whole other raft of other Nonpilus adhesins. We're not going to get too deep into those. So pili, remember, they're these long structures made up of a protein called pilin. Yes, it's very descriptive. And generally at the tip of them, they will have some sort of adhesion protein that can bind to cell surfaces. Maybe it binds to a receptor. Remember, pathogens are all about tricking cells. So maybe it looks like something that binds onto a receptor that's on the surface of a human cell. So these pili, we can think of them as little hooks for grabbing onto things. Here you can see pili attaching or linking E. coli to microvilli of the intestines. These are not necessarily pathogenic E. coli. So remember, we have a whole host of gut bacteria that help us digest our food and things like that. Here you can see the little pili attached to the cell surface of these villi, holding the bacteria in place. Otherwise, right, as things move through the intestines, these bacteria would be flushed out. And of course, some bacteria do get flushed out. Not all of them remain in place, right? Feces has large amounts of bacteria in it because of this. But, of course, some of them are going to stick around and hang on. We have two broad classes of pili. We call these type 1 pili or type 4 pili. Type 1 pili tend to be short, little hair-like appendages that are used only for just holding on. They're very static, they don't move or things like that. Type 4 pili are very interesting. They're more dynamic, they can move around, they're very thin and flexible, and they can result in what we call twitching motility, which you can think of like a grappling hook. So what happens is the bacteria extends its pili, which reach out and attach, and then it pulls itself along. So it throws out the grappling hook, grabs onto it, and pulls back in, moving along. This can be found in a broad range of gram-negatives. Vibrio cholera, which we've talked about before, does this. Pseudomonas aeruginosa, which can cause lung infections in cystic fibrosis patients. Neisseria gonorrhea does this. Sexually transmitted infection there. These are the two types of pili that are there. Of course, there are other things that can attach microbes to cell surfaces. We call these Nonpilus adhesins. These are generally proteins that are found on the cell surface, the cell wall, that bind to host proteins. Commonly, they'll bind to things like integrin or fibronectin, which are found on a lot of different cell surfaces. Here you can see all the little molecules on the surface of Streptococcus pyogens. And these are little molecules that will bind to the surface of host cells. Here you can see Bordetella pertussis, which causes whooping cough, bound to some hair-like cilia of a cell here. And it's doing this not through the pili, but through molecules that attach to and glue them to human cells. We're not going to go any deeper into non-pilus adhesins. There are so many different molecules that can do this, but just know we group them into two broad classes, pili, non-pili. So once bacteria have attached, then they go to work. And in this, they start replicating and dividing. So I'm going to attempt to draw a little picture here, and hopefully it will represent something like a diagram. Okay, so here we have a surface, right? And let's say it's the intestines, right? There's a bunch of cells here, and they're all along like the villi or something like that. So we have microbes that come in and attach to the surface here. And they do that by binding to the surface of the cells there. But once this surface is completely filled up, more microbes want to come in, but they have no cells to attach to. So they're just going to get flushed out, right? How do they stick around? Well, a lot of bacteria make what are called biofilms. So biofilms are, remember those extracellular polymeric substances? The microbial cells will secrete a sticky substance here, and the bacteria get trapped in there, and they grow inside of that, and that helps them stay attached to the surface here. So here we can see bacteria attaching to probably a gut surface or something like that. And you can see the individual bacterial cells in there, but then this goopy substance is holding them together in there. This not only helps bacteria stay attached, right, and avoid being flushed away or washed away or anything like that, but biofilms are also highly resistant to many chemicals, antibiotics. They hide microbes from the immune system. So biofilms are really worrying clinically. Biofilms, like I said, antibiotic resistant. So once you get a biofilm in there, you can try to apply antibiotics, but the bacteria are shielded from them. They can start secreting virulence factors. We haven't talked about this, but many microbes talk to one another and wait until they get to very high levels, and then they all start turning on their genes at once. They can hide from the white blood cells, the phagocytes, the antigen-presenting cells, things like that. Biofilms hide them. So there are a lot of chronic infections that involve biofilms. Oral infections, we talked about bacterial plaque that forms on the gums and teeth and things like that. That's a biofilm. Lung biofilms, very difficult to treat. Cystic fibrosis, patients with that, they often have biofilms of Pseudomonas aeruginosa that form in the lungs, and it can ultimately kill them. Urogenital infections, particularly when we have indwelling catheters or urinary catheters, things like that. Anytime we have an implanted medical device, biofilms can form on those substances, and once it's there, it's pretty much impossible to get rid of, particularly in elderly or immune-compromised patients. So they can stick around for years and cause chronic inflammation because they keep stimulating things like toll receptors and stuff. Catheters are the biggest one. I can't stress that enough. So many biofilms are found in catheters, and that leads to chronic infections. And as we know, urinary tract infections can be deadly, right? It's not just the discomfort of the urinary tract infection, particularly in elderly patients, that can quickly spread out into other regions of the body, ultimately potentially causing death. So a very worrying development that these biofilms occur on medical devices. Okay, so we're attached, we're growing. Now we'll look at the next part in chapter 18.3 when we look at how the microbes avoid the immune system and cause damage, get nutrients, things like that.